In every biology textbook, we are taught that we are a perfect 50-50 blend of our parents. One gene from mom, one from dad, working together in harmony. But there is a group of genes that refuses to follow the rules. For these genes, one parent's contribution is loud and clear, while the other is silenced, locked away by a chemical padlock before you were even born. This is genomic imprinting. Tonight, we explore the biological tug-of-war between your maternal and paternal heritage. Welcome back to my channel. I am the Geneticist. Today in Genealogy Guru, we are diving into the world of non-Mendelian inheritance. We've looked at the layers of epigenetics, but today we focus on parent of origin effects, where your body chooses a side. Genomic imprinting is an epigenetic process where only one allele of a gene is active, depending on which parent it was inherited from. While most of our 20,000 genes are expressed from both copies, about 100 to 200 genes are imprinted. These marks are established in the sperm or the egg and are maintained throughout your entire life, even after the rest of your genome is reset during early development. One of the most famous examples of this tug-of-war involves fetal growth. The gene IGF-2 promotes growth and is expressed only from the paternal allele. The maternal copy is silenced. Conversely, the gene H19 acts as a brake on growth and is expressed only from the maternal allele. This creates a perfect balance. The father's genes push for a larger, stronger offspring, while the mother's genes conserve resources to ensure her own survival and the health of future siblings. How does the cell know which side to silence? It uses imprinting control regions, ICRs. These are stretches of DNA that act like master switches. Using DNA methylation, a chemical off switch, and long non-coding RNAs, the cell can block access to a gene or interfere with its expression. In the case of the KCNQ1 gene, a maternal methylation mark allows the gene to be expressed, while the lack of that mark on the paternal side allows a silencer RNA to shut it down. Because we only rely on one active copy of these genes, there is no backup plan. If the active copy is damaged or if the imprinting marks are placed incorrectly, it leads to severe developmental disorders. For example, in a specific region of chromosome 15, if the paternal genes are missing, it results in Prader-Willi syndrome. If the maternal copy of the UBE3A gene is missing in the brain, it results in Angelman syndrome. These conditions are a powerful reminder of how vital it is that our maternal and paternal trait codes speak at exactly the right time. Your genealogy isn't just a list of ancestors. It's a carefully balanced conversation between the lineages that made you. One side speaks, the other listens, and in that balance, you find your health. If you want to see the gene-specific tables for IGF-2, SNRPN, UBE3A, and the mechanisms of methylation I discussed today, you can find the full research package on my Telegram channel. Thank you for joining me for this episode of Genealogy Guru. Stay curious, keep those cells dividing, and I'll see you in the next decode. See you soon.